time, I discovered a novel non-membranous particle that carry many proteins for constituent of centrioles and cilia. And you can see in the movie, the particle labeled by GFP moves along microtubules and accumulate to the centrosome. So by this study, I was really fascinated to the beauty of the cell. At that time, one of my dreams was to find a novel protein and to name it. And I found a new protein that exclusively localized to the tip of cilia. You can see this is tracheal epithelial cells, and there are many, many cilia from the one cell. And the green spot is the protein that exclusively localized to the tip of the cilia. And I named it sentan. Sentan means tip in Japanese. After that, I returned to dermatological field, and I joined Masa Amagai's laboratory and started the study of skin barrier. Why skin barrier? Because Professor Shoichiro Tsukita was a pioneer of tight junction study, and his laboratory identified first and second, third, fourth transmembrane protein of tight junction. And so he really opened the door of molecular biology of barriers. And in Japanese saying, a shop boy near the temple gate will recite sutras untaught. It means you learn without realizing it from what is around you. And for me, without realizing it, during 10 years in Tsukita's laboratory, I have become a specialist of tight junction. So my message to young researchers is you should be in a world best laboratory. And now I'm in Masa Amagai's, one of the world best laboratory, I believe. So skin is very beautiful and highly variegated among animals in reptiles, birds, and fishes, and mammals. And if you see fish skin, the scale of fish is completely different from our scale. Fish scale exists in the dermis, as you can see here in HE. And the epidermis, stratified epithelia, covers the surface of the fish scale. And there are many, many mucus-secreting cells in the epidermis of fish. There are no stratum corneum, only mucus covers the surface of the stratified epithelia in fish. In reptiles and birds, you can see very beautiful stratum corneum. And upper figure is the skin of reptiles. And by the way, the shell of reptile is a bone, deep bone. And this is the evolution of skin surface barrier in chordates. So in urochordate and cephalochordate, the epidermis is a single-layered epithelia, and the surface of the epithelia is covered by mucus. And when vertebrate appears, the epidermis becomes stratified, but still no stratum corneum, and mucus covers the surface. In fish and amphibian tadpoles, when vertebrate conquered the land, then stratum corneal barrier appeared. So you can see stratum corneal barrier in adult amphibian, reptiles, birds, and mammals. So the stratum corneal barrier is the barrier against desiccation to protect cells inside. But there is another very fundamental ancient barrier, that is the tight junction barrier. And all of these epithelia is barriered by tight junction. The problem to cover the body by epithelial cellular sheet is the leakage of solutes and proteins through paracellular pathway. To prohibit this leakage, tight junction barrier shields paracellular spaces. So tight junction barrier limits inside-out leakage of solutes and proteins, and also outside-in penetration of toxins and allergens. And stratum corneum barrier covers our body surface at the outside of tight junction barrier. So the problem to study tight junction is in vertical section, tight junction is only dots. So first we establish the three-dimensional observation way of tight junction barrier using whole mount staining of total entire epidermis of mouth ear skin. And you can see here beautiful honeycomb structure of tight junction. And each polygon is the cell of granular layer. And here you can see Langerhans cells exist below tight junction barrier. You can see the cell body of Langerhans cells in red and dendrites of Langerhans cells in blue. That is the Claudin one. Very interestingly, Langerhans cells express tight junction specific transmembrane protein Claudin 1 on entire cell surface, but they usually do not form tight junction. And we found in Langerhans cells in steady state exist below tight junction barrier, although they project their dendrites always in upward. But when once activated by the small irritation on stratum corneum, then activated Langerhans cells elongate the dendrite through tight junction barrier and reach to the bottom of stratum corneum. You can see in the right movie. What do they do? To visualize the antigen uptake, we topically applied tracers in green on the surface of mouth ear skin. Of course, there is a stratum corneal barrier, so this green tracer cannot penetrate into the skin easily. But there is some crack on stratum corneum, then the tracer can penetrate into the skin. Langerhans cells somehow activated by the crack of stratum corneum, and they elongate the dendrite to the outside tight junction barrier. Then the tight junction penetrated dendrite tip and penetrated tracers encountered at the outside of tight junction barrier. And you can see here the Langerhans cells taken up external antigens from the tip of their dendrite. And interestingly, Langerhans cells form tight junction barrier with surrounding keratinocytes. So they make a shield and uptake the external antigens. So when stratum corneal barrier is abrogated, then Langerhans cells activate and elongate the dendrite to the outside tight junction barrier and uptake external antigens, and they evoked Th2 immune responses. Very, very interestingly, Paul Langerhans himself had described that over than 100 years ago, dendrites end form a clear button-like structure and under the stratum corneal barrier between epithelia and conified layer. So left side picture is his sketch, and right side movie is our observation. And we are very, very surprised his sketch is so accurate, and you can see the typical Langerhans cells here. So some of dendrites for Langerhans observed, I believe they docked with tight junction. Then which layer of cells are equipped with tight junction? So the movie data is three-dimensional, so we can cut some place and can rotate 90 degrees. So this vertically aligned green line is tight junction. And when rotated in 90 degrees, tight junction is here. And upper blue is nuclei. So you can see basal layer nuclei in purple, and spinous layer nuclei in red, and granular cell layer nuclei in yellow. You can see stratum granulosum cells always triplet, 3 cells aligned vertically, and tight junction shields between second layer cells. So we call them SG1, SG2, and SG3 cells, and tight junction exists between SG2 cells. SG2 cells not only form tight junction, but also they have apical basolateral cell polarity. And this is human epidermis. And you can see only SG2 cells have green GP135 positive apical membrane and purple E-cadherin positive basolateral cell membrane. SG2 cells secrete lamellar bodies to form intracellular lipid lamella. And the SG2 cells secrete from through apical membrane. So intercellular lipid is filled around the SG1 cell. And SG1 cells itself become conified to make stratum corneum. So the layer of SG1 is very important, not only for Langerhans cells to uptake external antigens, but also to form stratum corneal barrier at the outside of tight junction barrier. And there is another beautiful structure in the skin. That is the, you can see here the 3 flattened layers, SG1, 2, 3 cells, and also you can see the corneocytes stacks very beautifully in vertical way. And you can see the zigzag pattern between corneocytes. And this 1, 2, 3, 4 is the like annular ring of tree. This is the record of the order of conification. So for example, number 2 cells locates upper than number 1 cell, and number 4 cell locates upper than number 3 cells, you can see. So this zigzag pattern is a record of the cell differentiation in 2 cell columns. And from this zigzag pattern, Alan and Potten in 1975 proposed the famous epidermal proliferation unit model. So they thought the cell division in basal layer is very regular, and that is the result. As a result, this beautiful zigzag pattern is formed. But recent live imaging showed the cell division in basal layer is much more random than expected. Then how such a beautiful zigzag pattern is produced? To make the long story short, the one key point is cell shape. And the scanning electron microscopic study have identified the flattened tetrachedron shape on the surface of the stratum corneum. So each corneocyte have this strange polyhedral shape. And we found that SG2 cells, tight junction forming cells, have identical flattened tetrachedron shape. And you can see in the movies, tight junction in green locates the edge of tetrachedron. what is tetrahedron? That is Kelvin's idea. And Kelvin's problem is how to fill space with cells of equal volume with minimizing the total cell surface area. And this is his answer, one of his answers, tetrahedron. And we can see this shape in nature, for example, in the seeds of pomegranate and in soap bubbles. So this movie shows flattened tetrahedron can fill space without gap, and tight junction is formed at the apical edge of tetrahedron cells. If this model is correct, then we can see variety of tight junction positive edge pattern. And so to show this, we isolated SG2 cells from mouse ear skin and stained tight junction. And here, this cell is an A-type cell, and all of the inner edge is positive in tight junction. And this is B cells. So one edge is outer edge, positive in tight junction, and other inner edge is positive in tight junction. And this is D-type cell. It's very funny. So outside, inside, outside edge, inside edge, outside edge, and the inside edge is positive in tight junction. And this is F-type cells. Entire outer edge is positive in tight junction. So all of the variation can be observed in vivo. And the question is how cells pass through tight junction barrier without abrogating the barrier. So only basal layer cells proliferate. So the differentiated cells moves upward and flattened in SG3 and gain tight junction in SG2 and lose tight junction in SG1. So cells continuously pass through tight junction barrier without abrogating the barrier. And we found some of significant percentage of tight junction polygon is double-edged. So most of the tight junction polygon is single-edged. This observation, from this observation, we think this model. So like an automatic door, double automatic door in the entrance of building. So when cell passes through tight junction barrier, temporarily double-edged barrier is formed like this and minimize the barrier leakage. So we established GO1 venous transgenic mice that is we can see in vivo live imaging the tight junction barrier in mouse skin. And here you can see sudden appearance of tight junction polygons. So in more detail, there is one tight junction polygon here, and every 30 minutes we observed, then new inner tight junction polygon appears, and tight junction barrier polygon become double-edged. Then the pre-existed outer older polygon become obscured and finally disappeared. During this process, the cell is at the starting point, the cell located at the inside of tight junction barrier, but tight junction barrier translocate to the top of the cell, to the bottom of the cell. Then this cell transfers to the outside of tight junction barrier. So the rule is very simple. In the flattened tetrahedron stacks, there are three different G-axis positions in one cell layer. And here, the cell higher than adjacent 6 cell is differentiated. So this yellow cell differentiated. And temporary double-edged polygon is formed here. Then the next, pink cells are higher than other cells. So pink cells is differentiated and translocated to the outside of tight junction barrier. Then next, blue cells are higher than adjacent 6 cells. So next highest blue cells are differentiated. During these translocation, every transition site, tight junction barrier, tight junction polygon become double-edged temporarily. So to limit the leakage of the barrier. And then the yellow cells again become highest and differentiated. If this model is correct, then it should be the size of the polygon is regular. So the in the double-edged polygon, outer order one is largest and newly appeared purple one is smallest. And surrounding 6 single-edged polygon should have middle size. Also, the outer green polygon locates highest in z-axis, and newly appeared purple polygon locates at the lowest, and the surrounding single-edged polygons locates middle in z-axis. So we return to in vivo data. And, no figures, sorry. So, this in vivo data confirms the location of tight junction barrier, and the size of tight junction is as predicted by the model. And we performed computer simulation here, and only this simple local rule, the highest cell around adjacent cells is allowed to differentiate, can explain how to maintain the homeostasis of barrier. So now yellow is highest, so differentiate, then pink cell differentiate, then blue is differentiate. So this simple local rule can govern the all of the epithelial cellular sheet to maintain the barrier homeostasis during cell turnover. So by this, in this way, if you choose the 2 cell columns, then always higher cells differentiate, then next higher cell differentiate. So always right, left, right, left, right, left. Such a regular pattern appears to differentiate. So I believe this is the answer how such regular zigzag pattern between corneal sites is produced. So it is the result to maintain tight junction barrier. And this is a very preliminary data, so we want to know how tetrachedron, 14-sided polyhedron appears. So this is the data from my collaborator, Dr. Nagayama, and he did computer simulation. And the rule is very simple. So if 2 cells become close, then repulsion force appears, and 2 cells become too far, then they become too much narrow. Then and simulate passage, then naturally 14-sided polyhedron appears in this computer simulation. So in this simulation, we set the parameter in the stratum corneum to compact in vertical way to make cells flattened. Then 14-sided hedron is the most popular polyhedron in this simulation. So it's really preliminary, and we need to study much more. But I believe the this cell shape is produced by the interplay between cell-cell adhesion and cell cytoskeleton that compact the cell to make the force to compaction, for compaction. So I'd like to wrap up my talk. So I really thanks to my mentors and colleagues and collaborators. So, and especially I really thanks to Shoichiro Tsukita, and also really thanks to Masa Amagai. So always he supports me and encourage me and do everything. And also, I really thank to my colleagues. Without tons of their support, I cannot do anything. So really, I want to thank to every people surrounding me. So thank you very much for kind attention.